Hello, my name is Paige Byers, and I'm here to talk to you about phenylketonuria, or PKU. So what is PKU? It is an inherited disorder that causes increased levels of the essential amino acid called phenylalanine in the body due to an inability to break it down. The increased levels of phenylalanine can cause intellectual disabilities, brain damage, seizures, eczema, light sensitivity, musty odor development, hair loss, and fair skin and hair. The fair skin and hair is a result of melanin synthesis impairment, which is directly related to the phenylalanine and its breakdown. This symptom is more drastic in the Asian and African populations due to their darker skin color. What is phenylalanine? It is an essential amino acid that is received through diet. In other words, we do not make this amino acid naturally. This amino acid is present in high concentrations in milk and dairy products, meat, fish and chicken, eggs, and beans and nuts. This amino acid is converted to tyrosine by phenylalanine hydroxylase. The tyrosine is used to produce many things in the body, such as melanin, which was discussed earlier, and the hormones dopamine, epinephrine, and norepinephrine, as well as many other products not mentioned here. Just to continue on about what PKU is, it is also known as an amino acid metabolism disorder, and it is the most common type of this disorder. This means that the mechanism behind processing the phenylalanine is hindered in some way. As you can see in the image on the right, the primary pathway is for the phenylalanine hydroxylase, or PAH, to break down the phenylalanine into tyrosine, which is then used to build other things later on. So what is the phenylalanine hydroxylase actually doing? As you can see in the image, the PAH enzyme is taking a molecule of the phenylalanine and a cofactor called tetrahydrobiopterin, or BH4, and adding a hydroxyl group on the para position of the benzene ring to produce the tyrosine. So since we know that this disease is in some way related to the rate of the reaction and the PAH enzyme working, we need to look at what factors affect the rate of reaction for enzymes. One important consideration is the enzyme's environment. This includes the pH, temperature, and location in the body. If any of these are wrong for the PAH enzyme, it could denature or unfold and not work properly. Luckily, in this case, we know that PAH enzyme is a hepatic enzyme, meaning it's located in the liver, and unless trauma is seen, the environment should not change enough to denature the enzyme. The second thing to consider would be the presence of all the right ingredients for a successful reaction. In this case, the enzyme PAH itself is needed, plus the BH4 cofactor mentioned before, and phenylalanine. Since the disease itself is a result of increased levels of phenylalanine, there is no shortage of that reactant. Also, the concentration of PAH enzyme and BH4 is not the problem. Another option to consider is just the sheer number of molecules of phenylalanine in the system saturating the existing level of PAH enzymes. If all of the enzymes are busy breaking down molecules as fast as they can and more molecules are ingested before the enzymes can deplete the current levels, it could look like the enzymes are not working properly. This goes back to the burger flipping analogy. If I have a grill that fits 20 burgers and you give me 40 burgers to cook, I can still only cook 20 burgers at a time. There's no difference if you stack 40 burgers beside me or 400, I still can't cook any faster. If this were the problem with PKU, though, it could be corrected with smaller portions of food saturated with phenylalanine being ingested in one sitting. So unfortunately, this is still not the reason behind the rising levels of phenylalanine seen in PKU patients. The last thing to consider would be a problem in the folding of the PAH enzyme itself or a structural error with it. One of these errors could occur during translation or transcription. Translation takes the mRNA and matches the 3 base codons to 3 that match for an amino acid on a tRNA to build a polypeptide chain. Transcription occurs when the mRNA is created from the DNA blueprint. Both of these processes have a possibility of error in one or more of the nucleic acid bases. However, if there was just one error in one mRNA or tRNA, it would not be a systemic problem, just an isolated one. So this leaves the only option being an error in the genetic code itself. meaning there is a mutation in the organism, or patient's, in this case, DNA. The error in the DNA code itself results in a protein that has an incorrect structure, and since enzyme activity relies on its structure, one change can cause large adverse effects in the enzyme function. So the case of PKU is a genetic mutation of the PAH gene. To date, more than 500 mutations in the PAH gene have been found or identified in patients with PKU. The location of the gene is located on the right at the yellow arrow. It is on the Q arm of chromosome 12 at the 23.2 position. A quick side note about this gene is that it is autosomal recessive, which means that both of the patient's copies of chromosome 12 need to have the mutation, or the gene would not be present, and the patient would not show signs of PKU. However, if you only have one copy of this gene, then you are a carrier and can possibly pass on the PKU gene to your children. To understand how a DNA error can occur, you need to first understand the structure of DNA itself. So DNA is composed of two strands of nucleic acids twisted around one another in a helix shape. This can be seen in the image to the right. The nucleic acids are composed of a backbone made of a sugar. In DNA's case, it is deoxyribose. With RNA, it's called ribose. And a phosphate group. Attached to each molecule of sugar and phosphate is a nitrogenous base. These can be seen as the letters in the image to the right. There are 4 nitrogenous bases: cytosine, adenine, thymine, and guanine. Adenine will only bind to thymine, and cytosine will only bind to guanine. The bond between all of these nitrogenous bases is hydrogen bonding. These are the bases that make up the code for the genes, and the order that they appear determine how proteins are built and how the cell function as a whole. The DNA acts as a blueprint for all the buildable substances the cell and the overall body needs to survive. The mutations seen in the DNA code are highly regulated when a cell divides, but even with the regulations, sometimes the mutations slip through. The following are the different classifications of genetic mutations. Missense mutations change one DNA base pair and result in the substitution of one amino acid in the protein it codes for. Nonsense mutation also change one DNA base pair, but this mutation results in a premature stop codon being coded for. The premature stop codon then shortens the protein and usually makes it unusable. Insertion mutations change the number of base pairs in a gene by adding DNA. Deletion mutations change the number of base pairs in a gene by deleting the DNA. Duplication mutation occurs when a sequence of DNA is abnormally copied. And lastly, a frame shift mutation is when an insertion or deletion changes the gene's reading frame. The reading frame is the 3 codon region that matches up with the tRNA for amino acids in the ribosome during translation. The mutation seen with PKU is missense. Most, but not all, of the mutations are based on the single base pair change associated with the missense mutation. Often, the mutation causes an arginine to be replaced with a tryptophan at the 408 position of PAH. Arginine is a positively charged amino acid, and changing it to an aromatic amino acid could cause changes in the structure and electrochemical interactions present during both the folding of the protein and the phenylalanine breakdown reaction. Other mutations linked with PKU and the PAH gene include deletions. Both of these types can result in enzymes that have reduced function or no function at all. So, how is PKU diagnosed? PKU screening is included in the slurry of newborn blood screenings. During these blood tests, the levels of phenylalanine are checked. This is done using chromatography or tandem mass spectrometry. In the table to the right, you can see the recommended blood levels of phenylalanine for newborns in the US. It is less than 60 millimoles. As long as the infant is in the OK levels, the baby isn't put on a low phenylalanine diet. If they do have worrying levels, though, the diet is the first way to treat the patient to prevent permanent brain damage and the other symptoms listed before. Currently, there is no cure for PKU. However, the symptoms can be treated and avoided completely if caught early and the treatments are stuck with. The dietary treatment is still the most common way to treat PKU. These patients follow guidelines like those that are shown to the right in order to keep their protein intake low, to avoid the symptoms that were discussed before. Often these patients eat large quantities of fruits, non-starchy vegetables, and special ordered low protein items. One experimental treatment is available for adult patients who have trouble sticking to the diet laid out for them. This treatment involves flooding the body with large neutral amino acids, such as tyrosine, tryptophan, and other branched-chain amino acids. The reasoning for this is because the large neutral amino acids share the same amino acid transport system across the blood-brain barrier. So if the transport is saturated with tryptophan or tyrosine, then less phenylalanine will be able to get across to the brain and damage the tissues. This is due to the direct competition between those molecules. This treatment is still in development and does not have the data to back up if it actually works well yet. So the best treatment is still dietary restrictions. The last therapy is for an abnormal form of PKU that affects the BH4, not the PAH enzyme. This abnormal form has a genetic mutation in the BH4, which is the cofactor involved in the reaction to turn phenylalanine to tyrosine. This mutation hinders the reaction, and this treatment is just to introduce healthy BH4 molecules, which would allow the reaction to proceed forward. For future treatment options, biochemists and genetic engineers may look into gene therapies to replace the underfunctioning PAH enzymes. Also, genetic therapy has the possibility of curing the disease if the mutations are identified fully and are able to be changed in the patient's genome. I hope you found this video to be helpful, and thank you for watching.